Airborne DNA reveals Arctic mosses are adapting rapidly to climate warming. New research suggests that the biological clocks of Arctic plants are accelerating. By analyzing decades of environmental DNA, eDNA, captured from the air, scientists have discovered that bryophytes, a group of non-vascular plants including mosses, are shifting their reproductive cycles significantly earlier in response to rising temperatures. The power of airborne DNA traditionally, tracking the seasonal movements of plants requires direct observation or ground-based sampling, which can be labor-intensive and limited in scope. However, researchers are now leveraging a more efficient method: environmental DNA, eDNA. Because most bryophytes rely on wind to disperse their spores, these microscopic particles become airborne, much like pollen from trees. By analyzing air filters, scientists can read the genetic signatures of the plants present in the atmosphere at any given time. This allows them to reconstruct a high-resolution timeline of when different species are actively reproducing. Decades of data. A Swedish time capsule, the study utilized a unique dataset from the Swedish Biodiversity in Time and Space project. Researchers analyzed 380 air filter samples collected at a monitoring station in Kiruna, northern Sweden, spanning from 1974 to 2008. By sequencing the DNA found in these filters, the team was able to identify 16 different genera of bryophytes. This long-term data set provided a rare opportunity to observe how these organisms reacted to a warming environment over more than 3 decades. During this period, the average temperature in Kiruna rose by approximately 1.7 degrees C. Accelerated reproductive cycles, the findings reveal a striking trend. The timing of spore release is shifting earlier in the year. The researchers noted several key changes by the end of the study period. Earlier starts. Most bryophyte groups began their sporulation season an average of 4 weeks earlier than in previous decades. Shifting midpoints. The middle of the reproductive season advanced by 4 to 7 weeks. Extended seasons. While the start dates moved earlier, the end dates varied by species, with some taxa experiencing significantly longer reproductive windows. Why is this happening? The shift is likely driven by a combination of thermal and hydrological factors. Warmer temperatures may allow plants to develop more extensively before winter hibernation, giving them a head start in the spring. Additionally, warmer springs lead to faster snowmelt and less snow cover, exposing vegetation earlier and allowing wind to disperse spores sooner. Bryophytes versus vascular plants. Perhaps the most significant takeaway is the speed of this adaptation. While previous studies have shown that vascular plants, like flowering plants and trees, in Arctic regions are advancing their flowering by about 2 days per decade, bryophytes are moving much faster. The data indicates that bryophyte sporulation is advancing by approximately 8 days every decade. This suggests that non-vascular plants may be more sensitive to or more rapidly reacting to the shifting climate in the far north. This rapid shift in phenology, the timing of biological events, highlights how sensitive Arctic ecosystems are to even minor temperature fluctuations. Conclusion: the study demonstrates that bryophytes are undergoing rapid reproductive shifts, advancing their life cycles much faster than many vascular plants. This acceleration serves as a critical indicator of how climate change is fundamentally reorganizing the timing of life in Arctic ecosystems.